continuing on that vein, it's indeed my proud privilege to invite Dr. Alok Srivastava, who is a legend in the field of hematology in India. He did his MD from the University of Madras and his postgraduate training at CMC Vellore. And from there on, he has acquired many more degrees. He was predominantly, he was a fellow at the Royal Australasian College of Physicians, and after that, Royal College of Pathologists Australasia and Royal College of Physicians UK. He has been an eminent teacher. He has been a founder of the DM Hematology program at CMC Vellore, which has trained a generation and future generations of hematologists in this country. He is a teacher par excellence. He is a giant in the field of hematology in our country. And now he has shifted his focus from transplant into the gene therapy area, and he is working on gene therapy for sickle cell disease and thalassemia and hemophilia. And I'd like to call Dr. Alok Srivastava to speak on his experience of the challenges in clinical trials for these therapies. So.
Thank you, Dr. Narula, and good evening, everybody. I want to also thank the organizers for the opportunity to participate in this interesting meeting, the likes of which I have not attended before, so it's something, a new experience for me. So being mostly industry focused and the title, the way it's been framed, I'm actually going to concentrate more towards product development and the challenges there, using the clinical and laboratory data only to focus on that. So, okay, so I'm going to narrate, first our experience with trying to take to clinical trial gene therapy for hemophilia B using an AAV vector, gene therapy for hemophilia A using a lentiviral vector. I'm going to give you a catalog of some of the other products that have completed their proof of concept studies and are ready to move into product development. And then finally, make a mention of the limitations and challenges in India, particularly with regard to product development and clinical trials, some of which may further get discussed during the panel discussion. So starting with hemophilia, as some of you may know, it is a deficiency of factor 8 or 9 that leads to bleeding. And for more than 50 years now, the treatment has been to replace protein, and more recently in the last decade, achieve hemostasis through other factor products. The bottom line is that these have major side effects and are also extremely expensive, the result of which has been that the vast majority of children and adults with hemophilia in India have been most inadequately treated. So recognizing this, many years ago, I had initiated a registry in the country which has now documented more than 22,000 patients, about 10,000 of whom have severe disease and therefore require regular replacement therapy, which is not available to them. But the advocacy for this has been so strong that this is probably the only condition where in more than 20 states in this country, there's a line item in the state budget for this disease. To the fact that about 500 to 600 crores, not a big deal in US dollars, but something $75 to $100 million every year are allocated for products for treating hemophilia in this country. And that still is highly inadequate because the requirement may be anywhere from 5 to 10 times that amount, which could take up a significant proportion of our health budget at the state or the national level. So realizing all this more than 20 years ago, I had started thinking about gene therapy for this. And this table gives you the list of clinical trials that were initiated in the late 90s, early 2000. The last one there is the one that was being conducted by a biotech company from San Francisco. And they actually had only Stanford and CHOP as the two sites for their therapy. And then they came and approached us to be the only third site in the world for their trial. We actually filed for registration with the regulator here at that time. And 6 months after that, the trial stopped because the third patient, an Australian physician, got expression but lost that expression in 6 weeks or so. And then, of course, people realized that the immunology in humans were very different from immunology in the mouse and dog. And nature had a little advice for gene therapists to go and learn immunologists with Ksy shows even now. So it kind of went into a hiatus for about 4 or 5 years. And then came this first successful gene therapy for hemophilia in 2011. This was a collaboration between the University College London and St. Jude, and a follow-up of 4 more patients, a total of 10 patients reported in 2014. Using the previous failed trial was AAV2 vector, this time it was AAV8, and all the ones who got higher dosage could show expression sustained, and this was a transformational study that then opened up the concept of how gene therapy could be successfully done with AAV in hemophilia. Now, we started talking about this, and it's been my privilege to know people around the world, and so I could bring the AAV biology from University of Florida at Alok Srivastava lab with the Emory University, Trent Spencer and his group to provide the transgene, and we put that together to say, okay, now we have a unique transgene that has many similarities with the ones that went into the New England Journal trial, but had differences with regard to the serotype, AAV3, the ITR, the promoter, as well as the codon optimization strategy. I'm not getting into the details of that, but that was then tested in 2 models. What's shown on the left is, AAV3 does not infect mouse tissue. So you need to put this transgene into an AAV8 and test that into the mouse, in the hemophilic mouse model, which is shown on the left. And on the right, you have a humanized mouse, a nog mouse, where about 70% of the hepatocytes are now human. And now you can put this transgene into the AAV3, which will infect the humanized liver. And what you see there is a dose-dependent response. Now, based on this, almost 5-6 years ago, we were ready to go to clinical trial. And the Powell Gene Therapy Center at the University of Florida, that had prepared 8 IND products, failed to produce AAV3 in appropriate titers for us to go to clinical trial. And that's where we got stuck, and we've been stuck there for the last 4 or 5 years, trying to shop around the world to see who can make this AAV vector for us. Finally, it's come down to making this vector in India, and I'm glad to say that we have developed an industry collaboration where hopefully we will see this happening in the very near future. But that's the failed story, so to say, of proof of concept studies completed but did not go to a clinical trial, because till today there is no vector making capacity for gene therapy in India. Thankfully we are just about developing the capacity for CAR T cells, which is a good thing, but not for others at the moment. Moving now, from there, so as was mentioned, we have 2 products registered, one about 7 or 8 months ago. This is $2.1 million. And if you thought 2.8 was, too much, just see this. This is the factor 9 gene therapy product that the US, the US doesn't fail to surprise us with the kind of cost they approve. So this is now $3.5 million. So, this is, there are ways you can calculate to these things. Now, AAV is an easy vector to use because non-pathogenic. It's an outpatient peripheral vein infusion, and your gene therapy is done. But there are challenges. The good part is it is safe, it is easy to administer, and so far, even on the failed trials of the early 2000s with nearly 20 years of follow-up, there are no major toxicities. But the challenge is in twofold. One, that this is ubiquitously present. And in this room in India, most probably all of us being adults, more than 80% of people have already been infected with AAV and have naturally occurring neutralizing antibodies, because of which you will not be eligible for gene therapy. And I'll show you some of our data that because of that, we then can't treat children, because this is liver directed, and unless the liver has enlarged to the adult size, people are avoiding using AAV therapy. So for these 2 reasons, even if we develop an AAV product, the number of people who will be eligible to be treated by that product wouldn't be very high. We continue our efforts in that direction, but many years ago, almost 10 years ago, we started thinking about alternatives. This is probably the most extensive study done for AAV serology in the world, with nearly 1,000 people participating. What I want you to see is that by the time you are an adult in India, you have 80 to 95% chance of being anti-AAV positive. And that is not limited to 1 serology, 3, 5, and 8. All the ones which are most commonly being used in clinical trials show similar profile. 3 is a little bit less, so we have some hope, but we know that there will be a lot of exclusions because of this. So based on this, we started collaborating with the Emory University about more than 10 years ago, where we thought we would put the factor 8 gene into a hematopoietic stem cells and under a promoter that is lineage specific, and then see how it would do. So this has gone on now to become a product for a clinical trial. That's the construct where you see a promoter which is CD68 and monocytic lineage specific. We have a specially designed factor VIII construct that gives high expression in the monocytic cell, and the product then is a CD68 factor VIII construct in the lentiviral virus, which then is used to transduce the autologous hematopoietic stem cell. This was tested in 2 mouse models. First, the hematopoietic stem cell of the mouse in the hemophilic mouse to show that it had adequate expression, and there actually it reached almost 100% expression. Then it was tested in the NSG mouse with the human hematopoietic stem cell, and what you see on the right is still significant expression. Based on this data, the FDA gave approval for this trial in 30 days in 2019. We applied in 2018, and I'll show you the chronology. But we were able to, this product was manufactured. They also had challenges. It took about 3 years for them to sort out this lentiviral vector production. They tried it themselves, it didn't work. They went to a company that made two batches, and that company got bought up by a bigger company, so those 2 batches went into, they couldn't use that data. They had then to go to the Cincinnati Children and finally made their lentiviral vector now, and this is what we are using in our clinical trial now in India. So, I'm glad that in the previous talk you were told how this is done. So that's what we do. We do an apheresis, we collect the stem cells, and then we do the transduction in our lab. We assess the quality, particularly the vector copy number, and then if everything is fine, then we go ahead and do an autologous transplant with those transduced cells. We don't use busulfan for a long time now. We have been using triosulfan, which is certainly reduced toxicity with myeloablation. Unfortunately, triosulfan is not available in the US, so they are forced to use busulfan. You can take it from India. We actually manufacture it here. Now, I wanted to show you this because this is a chronology of how this application went. So it took about 3.5, 4 years for it to go through the CDSCO, from the CDSCO to the CBBD Tech, from the CBBD Tech to the RCGM. The RCGM did not think they had the capacity to evaluate this. They appointed a special committee. Then that special committee met. Finally, we had 2 presentations with them. Then they gave it back to the CDSCO. It went to the SEC 3 times after that. 3.5 years. So, if it was truly a sponsored trial, I don't know what would have happened to the company, but thankfully in academia, you still have your job while this is going on. So, that's for hemophilia. That's where we are. So we are doing the first in human lentiviral mediated hematopoietic stem cell based gene therapy for hemophilia A at the moment, and 2 patients have been treated. Moving very quickly to the major hemoglobin disorders, which is a major public health problem. Here it's not academic interest. So these are some statistics. You have seen some of these earlier already. In this country, we have large numbers of people being born with no effective control program. So creating an effective control program is another goal. We are working on that with the Odisha government in Odisha to create a model for control of thalassemia and sickle cell disease in the country. But on the curative end, as you know, our center has been doing transplants since 1991. We've done more than 700 transplants, probably one of the largest series in the world. But we also recognize that for 700 transplants that we have done, hundreds of thousands of children have been born here, and even if we reduce that through a control program, curative therapies are needed for all the reasons that was pointed out in the previous talk. So towards that, as you know, there are 2 approaches, at least at the moment. You take the hematopoietic stem cells from the patient, you can do gene modification using a vector, you can gene add, you can add a beta or a gamma globin gene, or you can gene modify using an shRNA approach through a lentivector, or you can gene edit. You can use a CRISPR-Cas9 approach, you can use a base editing approach, and you can also do gene correction with a prime editing approach. And I'll show you that our catalog includes all of them, and most of them have completed proof of concept studies to take it forward. So this was the data based on which the lentiglobin was approved in the US. I'll not go into the details, and as was mentioned already, the cost is $2.8 million. Now this is our first gene. So 𝑅𝐨𝐥𝐺 𝘠𝑦 in our group has been interested in hemoglobin biology for more than 2 decades. He works on developing gene addition vectors. What is shown here are both beta and gamma globin addition vectors, which we have developed in collaboration with Trent Spencer's group at the Emory University. This data looks good enough to go to the trials. But when you have a choice, you decide which one to take forward. So this exists, and I won't, as I said, I won't explain the data, but it will give you an idea of what's being done. We also are aware of the shRNA-based approach to increase hemoglobin F, the basis of which was explained earlier. This is the first 6 patients where you see good response to using BCL11A modification through an shRNA lentiviral vector-based. And this is exactly what Shaji has developed. So this is actually the first product that we want to take to the trial, where we have our own target for this shRNA-based approach for beta thalassemia and sickle cell disease. And this is what we hope to take into the clinic for this trial. Now, when it comes to gene editing, you have different things you can do. What is shown in this cartoon is that you can target the BCL11A at the erythroid enhancer aspect, which is A. You can do B, which is below that, introduce a non-deletional HPFH, create a point mutation. You can introduce a deletional HPFH because deletions like it occurs naturally in nature. And finally, you can do the gene repair with something like prime editing. So 2 scientists in our group are working on this, and I'll show you what they've achieved. This was the paper published about 2 years ago, where with just 2 patients, one with thalassemia major and the other with sickle cell disease, they could do the correction using the CRISPR-Cas9 approach, and showed that there was significant elevation of hemoglobin F to the point that the disease phenotype was completely ameliorated. So similar to this, Sarvana Bhavan in our group has found unique targets which he is able to modify and create a deletional HPFH-like phenotype. And he has completed these proof of concept studies. So we have we have an in vitro model where we have an ex vivo erythropoiesis where this is tested. And after that, we have the in vivo model in the NBSGW mouse, not just the NSG mouse, but the NBSGW mouse, where you get much better erythroid differentiation, unlike the NSG mouse. So in these 2 models, these have been corrected, and we are looking for taking this again into the clinic. This is ready for that. Now, we also have Mohan Kumar, who works on base editing and prime editing. So he has identified specific targets in the BCL11A enhancer region, as was shown earlier. And what you see here is a very high degree of efficiency in gene editing. You can see about 80-90% with the 2 targets in the right. There's no pointer here for me to move. And that translates to significant elevation of hemoglobin F. So this is actually in discussion right now with one of our industry collaborators to see how this can go forward into a clinical trial. What he did want me to show also was something that he published last month. Gene correction using prime editing. It was mentioned in the last talk that this is something that is just happening, not easy to do. Well, this has, and you can see that there is a significant efficiency with which Mohan's lab is able to achieve this. So this has to go through a few more studies on the proof of concept, but this is again something very interesting that we are in the process of developing towards gene therapy for the beta hemoglobinopathies. So we are all aware, we are all in a hematology department where we do hematopoietic stem cell transplant, and once in a while you land up with a donor who has certain phenotype which gives you unexpected results. So people with HIV infection, when they were transplanted with a particularly naturally modified CCR5 receptor, it turned out they were also cured of their HIV because this receptor becomes resistant to HIV infection. So then can we take people's autologous stem cells, modify the CCR5 and give it back to them as a gene therapy for HIV infection? So this is not new. People are attempting this already in different labs. This is what Sarvana Bhavan has actually achieved. He has managed to achieve CCR5 disruption with great degree of efficiency. We have collaborated with our virology department to show that there is significant resistance to HIV once the CCR5 has been disrupted in the hematopoietic stem cell. He also has some techniques of using the more long-term HSCs. We purify from the HSPCs, the long-term HSCs, and modify only those, and that has certain advantage towards reducing cost and complexity. So I wanted to finish with this slide. We put this together about 12 years ago as part of the Clinical Translation Committee of the ISSCR, where we just kind of wanted to define the path to the clinic from proof of concept studies for cell therapies in general, because a lot of abuse of cell therapy was happening in the community at that time. But this formed a good basis for us to discuss today where the challenges are in our country. So first we have the challenge at the level of not enough academic units and almost zero industrial units really doing basic research towards the proof of concept studies. So that's a challenge here, and how to solve that is a matter of a great discussion. The second big area is that even if a few labs do something, the interest from industry to get into this space, which is much more nebulous in terms of the business model and the returns, is so unsure that at least in India, there are very few who are daring to do that. But I'm glad to say that it's not zero. So it's very good that it's happening. But in that process, there are many reagents, growth factors, cytokines, mRNA, Cas9 proteins, which also need to be produced locally at the GMP grade, without which this will not happen. And if we have to buy those from manufacturers overseas, then the cost cannot be brought down. And then finally, the regulators cannot take 3.5 years to approve. Hopefully that was the first time, but going forward, it'll be much better. So with that, I want to acknowledge our team in hematology, who is involved who are involved with the clinical trial, a group at the Center for Stem Cell Research, a completely Department of Biotechnology, Government of India funded entity, and the people who work there, a long-term collaboration with colleagues at the Emory University as well as the University of Florida. None of this would be possible without the generous funding from the Department of Biotechnology over the last 15 years, and also for the clinical trial, we have partial funding from the Emory University. And also the patients and their families for their trust they place in us to go through a first-in-human. I can tell you we've done many clinical trials, but the consenting for the first-in-human trial with a lentiviral vector is not the same. So with that, I want to thank you for your attention and leave you with this thought.